Okay, let's continue. Play the video.
Basis of the bacterial from the glycolysis pathway from the normal well-known E. coli. So different bacteria actually have different glycolysis pathway. Okay, so this you have to bear this in mind. So we know the growth phases of the bacterial growth, but practically how we measure the bacterial growth. Okay. I introduce two common ways. The first one is plate count. Plate count is a direct method to measure bacterial growth. Then you directly count how many cells grow, okay, on the agar plate. And the turbidity normally used for the growth, okay, liquid condition. And you check the turbidity of the cell culture in order to measure the bacterial growth. This is indirect method. You don't really count the cell numbers. So for the plate count methods, you actually count the colonies on the plates that have less than 250. Okay, between 25 to 250. You don't count the plates has too little cell. You don't count the cell with too many counts because they are not accurate. We call the colony number called colony forming units, abbreviated as CFU. So you see CFU quite often in the textbook or papers because this is how we count the bacteria directly from the agar point. The way how we did is we actually dilute the cell suspension using a tenfold dilution. So this is a serial tenfold dilution in the test tube. And we take an aliquot from the cell suspension and plate on the agar plate. And this is too many bacteria, so we cannot count. So we will count approximately this range, and then we will average the cell numbers by different dilution, okay, and then to get the cell numbers, okay. Because the reason to use colony forming unit is using the particular media, for example, in this media, maybe some other, if this is more than one types of species, we don't know exactly whether all the bacteria can grow well on this particular media. We can only count how many cells to form colonies, okay, on the plate. As for the indirect method called turbidity, normally we use spectrophotometry. Okay. The turbidity is this principle is shown here. So this is the cubate containing the cell suspension. If the bacterial grows, this is if there's no growth, all the lights will pass through. So the transmission is 100% as a control. So during the bacterial growth, the turbidity increase. So the transmitted lights will be decreased. So we use this as indirect method to measure the cell growth. Okay. So now I introduce two types of normally used method to measure the bacterial growth. Turbidity is an indirect method. but practically it's the simplest way to measure growth. Because if you use a CFU, you need to grow up the cell. You can only know the cell number maybe overnight or on second day. For turbidity, you can measure the cell growth right away. Okay. So you need to have a standard curve for the individual bacteria as different cells, as different size, therefore has different transmission effect on the light. So what you should do is you should have cell numbers versus the optical density from the spectrophotometer. So here is the standard curve with the optical density OD versus cell number, okay, per mil. Okay, so the line derives from this at the linear range, okay, from different cell numbers. Therefore, you got a linear correlation between these cell numbers and the OD. Then you can use this as a standard curve for your particular cells. So as I mentioned, that some big cells, one may be equivalent to 3 smaller cells. One coccyte maybe is more than 3. One rod cell maybe is more than several coccus, right? So, but the cell number is 1 to 3. Right? So the cell number would be more accurate if you take advantage of the practical way such as stability, then you have to calibrate the cell number. Okay. So what happens if the cell density is too high? Okay. So some of the lights is rescheduled. And then therefore the numbers will be lower in the readings, in the optical density readings. So how to solve it? Meaning, when the cell number is too high, the correlation between the cell numbers and OD will not be at a linear range. But when you practically grow the cell number keep increase. Okay, so in order to best fit the standard curve at a linear range, normally we need to dilute the cell culture within the range. Therefore, we have the accurate cell numbers counted. Okay, so this is how we solve it. We actually will need to dilute highly dense cell culture in order to make the cell count more accurately.
In this video, you will learn the proper practices and procedures for performing aseptic technique. Using aseptic technique, you will be able to transfer a bacterial monoculture into a fresh medium without introducing any contaminating microorganisms. Two of the most commonly used tools for transferring bacteria are the inoculation loop and the inoculation needle. Knowing how to use a Bunsen burner is an important skill in aseptic technique. To light a Bunsen burner, start by closing the valve at the base by rotating the collar so that the vents are covered. Then slowly turn on the gas. Once you hear the gas, ignite the burner and set the height of the flame by adjusting the amount of gas. The temperature of the flame can be adjusted by rotating the collar to control the flow of oxygen. The coolest flame is yellow and orange. A medium flame is blue. And the hottest flame is a roaring blue flame characterized by a clear blue cone in the middle. The tip of the cone is the hottest part of the flame. Inoculation tools are sterilized by passing them through the hottest part of the flame. Once sterilized, inoculation tools should not be set down on any surface. In this section, you will learn how to transfer bacteria from a liquid culture to a petri dish. After flaming the loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid. Transfer the bacteria to your petri plate. Lift the lid of the plate and hold it over the base as a shield to prevent contaminants from falling onto the plate. Spread the bacteria over the entire surface of the plate in all directions. Rotating the plate a quarter turn between spreads will ensure that the bacteria is spread evenly across the plate. In this section, you will learn how to transfer bacteria from a liquid culture to a liquid. After sterilizing an inoculation loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid. Now take the tube of liquid media and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Flame the mouth of the tube before replacing the lid. Flame the loop to sterilize. In this section, you will learn how to transfer bacteria from a petri plate to a stab culture. Start by sterilizing an inoculation needle by passing it through a flame. Allow the needle to cool. You can test for coolness by tapping the needle in a clear area on your agar plate. While holding the lid as a shield, gently rub the inoculation needle over a single colony. Transfer the bacteria to a culture tube filled with nutrient agar. Remove the top of the tube and insert the needle, making 3 deep stabs. When finished, replace the lid on the tube and sterilize the needle. In this section, you will learn how to transfer bacteria from a petri plate to a liquid. Sterilize an inoculation loop by passing it through a flame. Let the loop cool. You can test for coolness by tapping the loop in a clear area on your agar plate. While holding the lid as a shield, gently rub the inoculation loop over a single colony. Now take the tube of liquid media, remove the lid, and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Pass the mouth of the container through the flame before replacing the lid. Sterilize the inoculation loop to complete the process. In this video, you will learn how to perform streak plate technique. The streak plate method allows isolation of a single colony from a bacterial culture by splitting the plate into quadrants and diluting the bacteria repeatedly as the loop is streaked through each quadrant. To light a Bunsen burner, start by closing the valve at the base by rotating the collar so that the vents are covered. Then slowly turn on the gas. Once you hear the gas, ignite the burner and set the height of the flame by adjusting the amount of gas. The temperature of the flame can be adjusted by rotating the collar to control the flow of oxygen. The coolest flame is yellow and orange. A medium flame is blue, and the hottest flame is a roaring blue flame characterized by a clear blue cone in the middle. The tip of the cone is the hottest part of the flame. Flame an inoculating loop to sterilize and allow it to cool. You can lightly touch the loop on an empty spot on the bacterial plate to make certain the loop is cool. Gently scrape a colony from the surface of the plate so that it collects on the loop. Transfer the bacteria to your LB auger plate and gently rub the loop back and forth across the top left corner of the plate about 10 times. Stay in the top left quadrant of the plate and do not break the surface of the auger. When finished, flame the loop. Rotate the plate 45 degrees or a quarter turn and draw the loop through one end of the first streak and rub back and forth in the second quadrant about 10 times. Flame the loop again to sterilize. Rotate the plate 45 degrees and draw the loop through one end of the second streak and rub the loop back and forth in the third quadrant about 10 times. When finished, flame the loop again. Repeat the process one final time in the fourth quadrant. Flame the loop after completing the streak plating. Invert the plate and place it in an incubator at 37 degrees Celsius for 24 to 48 hours. After 24 to 48 hours, the colonies will have grown and can be examined.
Okay, the following slides, I'm going to describe how we do the material culture and scale up at laboratory scale, I mean the basic research lab. So normally we plate on the cell, strict on the plate to get a single colony. As I mentioned that a single colony represents from one bacterial cell to grow up and accumulate to be a visible colony. With that, we can transfer into 5 ml broth in a test tube. Okay, so normally when we scale it up, we will use a ratio about 1 to the 50 or 1 to 100 dilutions. As this example, if we use 0.5 ml inoculate into the 50 ml broth, is 104 inoculant ratio. Or if it's 1 ml, it's 1 to the 50-fold dilutions. So another concept that we normally use is if we use a flask to cultivate bacteria in broth, the ratio of the broth versus the total volume of the flask is about 1 to 5. ratio. So normally we will not use like half of the volume of the cultural suspension in the flask, because I will talk about this later. And for the following scale up, for example, if we will transfer further into 500 ml broth, we will use again 1:50 ratios for the inoculant run. So the next scale in our lab is use 3 liter flask containing 500 ml broth. So it's about 1:6 volume ratio. Okay, so if we want to use a fermenter in the lab, so it's a 10 times increase in terms of the cultural volume. Then again, we use 100 ml of inoculant approximately. Of course, sometimes it varies depending on the concentration of the cells in the inoculant. In this slide, I'm using a photo to demonstrate how we do the scale up at laboratory scale. For example, this is the colony that I already described. It was stripped on a plate using the 4-phase streaking methods in order to get a single colonies from the agar point. And then one colony we will transfer into the 5 ml culture in a test tube, okay, with sufficient space in the test tube. This is for the aerobic bacteria. Okay, for the aerobic bacteria, normally, as I mentioned, that we will leave enough space for the bacterial suspensions in the flask. Okay, then with the 10-fold scale up, as I mentioned, 5 ml, 50 ml to 500 to 5 liters. Okay, so in the fermenter, the working volume 5 liter, normally the complete volume will be like 8 liter, because we still need some space above the suspension. Okay. For the aerobic bacterial cultivation, there's a strategy to increase dissolved oxygen. For the test tube, okay, we will have like 455-degree angle to incubate for overnight using E. coli as an example. And normally we use 200 RPM. As for the flask, this is a 2.8 liter Belco flask. It is relatively expensive compared to the normal flask. There are two unique properties. First one is the shape of the flask is more flat, and then it's a shorter. Therefore, you got a bigger area in the bottom. Secondary, it has a groove design, meaning that the suspension in a rotary shaker, the bacterial suspension will actually hit the groove, and there's more mixing and turbo happens during the rotary shaking growth. For example, it's 200 RPM for aerobic bacteria. That's normally the speed we use. So this is the strategies in flask, but this flask is actually quite expensive. Okay. On your right hand side is the fermenter. This is a 5 liter fermenter. Please pay attention to the baffle. Did you see this? 1, 2, 3, 4. There's 4 baffles inside the glass cylinder, which have the same function as a group. Okay, and in the center there's a motor connected to an agitator. When the agitator spin, it will mix the growth suspension inside the tank. And those baffles will help to mix and also to increase the oxygen, dissolve oxygen in the culture. Of course, there's a oxygen probe to monitor the oxygen concentration in the culture. As for the bioreactor used to scale up further